recording. Okay. Thank you very much, Dr. Denise Gross, for this third interview. Today, we will discuss about the next generation of COVID vaccines. But let's start by a question. What's open innovation for you? Someone told me, I read very recently an article about someone, an expert has written an article about BioNTech and Pfizer collaboration. And in that article, he claimed that this is a kind of open innovation. I can't see any open innovation in that type of collaboration because of the reasons that we have already discussed. So what's open innovation for you, and does it happen in biotech and vaccine industry?
I think less than biotech. I think you're right, partly because of the intellectual property. That for many of these startups, that's what we would call their family jewels.
Mm-hmm.
That's how they got started, and that's what keeps them in business, and that's also their platform
Mm-hmm.
of being able to continue their enterprise is having intellectual property they can license and use the
Mm-hmm.
cash flow from a license to keep going with the next generation or whatever their technology is.
Mm-hmm.
A good example, I worked with a company out in California, and they had a development platform for screening of cells, and they had the instrumentation. They also had a series of dyes that were used to look at transport of drugs into cells, and they made the first generation publicly available. So here's open innovation. Anybody that wanted to use it after they published their papers and released it to the public could use their materials. However, if you wanted to collaborate with them and develop a drug, their business model was that they would license their dyes to you that would run on their instruments. And part of the license agreement was that you would take a development program that you had and share it with them on their site. And if you discovered something or they discovered something, you would both share in the intellectual property and whatever would go forward. So, they licensed their technology to you, but they became a business partner with you and continuing to develop. So the first generation of their dyes was wonderful, but were not really all that good. They were good for academicians to use to get an initial signal. But if you wanted the good stuff, you have to enter into this business plan of transferring one, if not more, programs to their site and working with them collaboratively. So I think open innovation probably works well at the university level, where there's less, even with the Bayh-Dole Act, there's probably less pressures to patent. Uh, a lot of the incentives aren't quite there yet because, at least in the United States, and I don't know about France or the EU, a lot of schools still don't understand innovation, and
Mm-hmm.
faculty still aren't being rewarded
Mm-hmm.
for doing it. Their job is to teach. Their job is to sit on committees and develop the next generation of scientists or people in the humanities. And so innovation is not something that some schools value. And so, open innovation is more sharing, and that's something that industry doesn't do well because of a long history of patent protection,
Definitely.
either via patents or trade secrets, which
Mm-hmm.
allow you to have something that doesn't get patented. But as soon as
Mm-hmm.
you start talking about a trade secret, then it's no longer a trade secret, and then it's open to the public, and anybody can technically use it.
Mm-hmm.
So I mean, people talk about open innovation. I don't think we're there yet. Um, and what's even very worrisome is the whole concept of funding right now. I was just exchanging emails with the chairman of my board of directors, and I was talking about a report I just read last week from a financial analyst, and he was absolutely appalled about the current spate of drugs of companies, biotech companies especially, going public. Even if they're, say, a platform company and they've got a novel platform that they think other people can use to discover drugs, or if they actually have a drug in a space that is really important, and they've gotten 2 or 300 million dollars worth of venture capital funding. They've gotten to that stage, and now they're doing an IPO. And they're doing IPOs, and they have absolutely no clinical data whatsoever. It's just hype. And he's appalled by that. He said, 10 years ago, that wouldn't have happened.
Mm-hmm.
Somebody would have needed to see real data, not just a lot of hand waving. Trust me, it'll happen.
Mm-hmm.
Magical thinking, as Joan Didion called it in her autobiography a couple of years
Mm-hmm.
ago. And he's just appalled by the fact that that's happening, and the public is buying into it in
Mm-hmm.
some cases. So it's going to be interesting to see how this all pans out. And COVID may be the trigger. It's been both a plus and a negative for for biotech. Things, yes, move quickly. I know you wanted to talk about next generation vaccines. I think the next generation of vaccine may not be the solution to solving the COVID crisis. So, just 2 days ago, the U.S. government announced they're going to invest $3 billion in antivirals, not in the next
For
generation
treatments,
vaccine,
yeah, for
for treatment.
treatments. We
Or
need treatments, of
we
course we
need
need
treatments because,
treatments.
hey, I'm putting my global health hat on again. Getting vaccines to sub-Saharan Africa or to far-emerging countries, as we call them, is probably not a viable option for many of these because it's still a matter of how do you dispense them? How do you follow up? How do you keep track of the adverse experiences? How do you go back for that second dose in many populations? Antivirals could be, a handful of capsules that you give somebody or tablets that are probably, and I've pushed this for a while when we talk about treating diseases like this, there's probably a much better option. Now, the downside is there's only three ways that you can deal with in treating a virus. You have three mechanisms. You either prevent entry, you
Mhm,
prevent the virus from replicating, or you
mhm,
prevent the virus from leaving the cell.
mhm,
So while you don't have direct-to-consumer ads in Europe, in the United States, we still do. And so you see a lot of the newer drug combinations for HIV/AIDS, and they are combination products.
mhm,
They may partially prevent entry into the cell, and they may partially block biosynthesis of the next generation of viral particle before it leaves the cell. And that may be the solution because in many cases, some of these drugs for HIV have actually reduced viral load in the body to non-detectable levels,
mhm,
telling people you can have sex again
mhm.
and not worry about transmitting to either your partner or to, say, a fetus. So in the case of HIV, it's worked out amazingly well because we will probably never see a vaccine to HIV. It's because of the nature of the virus, how quickly it evolves, how quickly it mutates, how quickly it infects the cells. Plus, what it infects is not the cells in your body; it infects your immune system cells,
Mm.
and so that's difficult to deal with. But for diseases like COVID-19, SARS-CoV-2, or whatever's coming next, Marburg virus, if that resurges, or any of the other diseases out there. I have a chart I'll send you of what they call emerging diseases and re-emerging diseases
Mhm.
that we
Yeah,
need to worry about. We're talking
definitely.
about dozens and dozens and dozens of diseases out there, and having something maybe that goes across multiple types of viruses. And
Sure.
there's a
Mm.
company here at Doylestown that's working on that because they found a platform, they have found an enzymatic mechanism they think applies to multiple diseases.
Yeah.
And that may be the secret of being able to rapidly
Mhm.
deploy something in the far emerging countries and in countries where we have good distribution technology, such as the EU or the United States or Canada.
I have still a lot of questions about the
Sure,
platforms I wanted to ask you. Yeah, I
sure,
wanted
sure.
my question about the next generation vaccines. It was about regarding Novavax. You know that this week Novavax announced very good results with its
Yep.
mRNA vaccine. You know, and I just read this evening. You know, an article, a short. It was a short article written by one of the most famous European scientists. He lives in Brussels. He wrote, you know, what are the considerations, what are the lessons that we can learn from CureVac, you know, failure. CureVac this week, you know, announced its almost failure, and Novavax success, also Pfizer BioNTech success, and what can be the next generation of the vaccines. I have read a lot this week about BioNTech platforms, technology platforms. One of my questions, and also about the history of their activities, I saw that they have never sent any treatment to the market. So they
Nope.
work, they
Nor
work,
has Moderna.
yeah, they work only on platform. This has become a secret for me. What are these platforms? Where they do come from? How do they create? You know, what's a platform? Can you explain for us, please?
Well, the platform would be like that messenger RNA technology of using
Yes.
that and then as a vehicle. The same thing with the J&J platform, which was
Mhm,
that AAV vector, which was originally developed for gene therapy to
mhm,
get something into the cell and get it into the cell. And that's always been the issue. So it just isn't the messenger RNA technology that was important. It was the vehicle,
mhm,
those lipid nanoparticles.
mhm.
If you just stick messenger RNA into your arm, it ain't going anywhere. It's
So
not going to
it's
do
only
anything.
theoretical science, yeah?
Well, it's not so much theoretical. You're right. From one standpoint, they've been working on it for probably 10 or 11 years of figuring out how do you get that messenger RNA to code for something that's important. So in this case, it was the spike proteins,
Mhm, mhm,
which then would let your body mount an immune response to just the spike proteins
mhm,
without having to have anything present. And that worked out well because but people had tried that with HIV. Now, the problem with a vaccine for HIV is it doesn't have a lot of surface geography to deal with. You don't have these spikes sitting out there. You can't take the spike off of HIV because it doesn't exist. You're looking for other things on it. So this has worked out well, but it was a matter of could you create enough of a message that the body
Yeah.
would respond to. And in this case, it was the spike proteins. So
Yeah.
that was our platform was can we make a messenger RNA that would code for just enough of a gene sequence to make enough of a protein that the body would then respond to.
Mhm.
And then it came down to what's how do you find an appropriate animal model?
Mhm.
Because when you have to do safety toxicology safety studies on any of these materials, at least in the United States, the Food and Drug Administration and the EMA is going to say you've got to go to a relevant animal model, which means finding an animal model that is going to respond like you and I would.
Mhm.
So everybody falls in love with mice, but people tried that for a long time for influenza. Mice don't get the flu.
Mhm.
Ferrets get the flu. So you need to have, populations of animals that are appropriate that will let you do the safety studies. And it
Mhm.
ultimately came down to looking at small primates, cynomolgus monkeys, to find out do you really get a good immune response. So that's why I think Pfizer originally signed their material transfer agreement, their memorandum of understanding with BioNTech, which, as we talked about last week, was was for the flu because you need something that you can do rapidly. You can't
Mhm.
spend 10 months making the next year's flu vaccine and find out that two of the three strains that you thought you picked properly with the World Health based on
Mhm.
activity in the southern hemisphere in Australia and New Zealand turned out to have mutated by the time they got up to the northern hemisphere.
Mhm.
And for many, many diseases, they are seasonal. You're not going to test a flu vaccine when it isn't flu season.
Mhm.
And so that becomes a rate limiting step. So, the hope would be with something like that messenger RNA platform that you could do something, plus the speed. But it was also figuring out how do you get that messenger RNA into a delivery vehicle.
Mhm.
In this case, it was those lipid nanoparticles. Yes, that's interesting technology, but it's not new technology. There's been a company in the United States for years called the Liposome Company,
Mhm.
and their mantra was, trying to figure out, excuse me, how do you get drugs into lipid nanoparticles that you could then get to a site and
Mhm.
not have them circulate widely in the body to reduce side effects. If you've got something that's really toxic, a compound for cancer, or a
Mhm.
very, very toxic
Mhm.
antibiotic, you don't want it
Mhm.
floating around everywhere. You want to target it.
Mhm.
And so that I think was part of it. It was really, part A was the messenger RNA, but also part B was making that lipid nanoparticle. And Moderna was in the same kind of a position of being able to figure out how do you mass produce it? How do you get it so that it encapsulates the messenger RNA
Mhm.
and you have enough load in a vial of encapsulated messenger RNA to be able to say, here's our load when you inject it into somebody. Here's how many copies of that messenger RNA are there, because that's the only way it's going to be protected is inside that lipid nanoparticle. So that formulation issue is also something that people neglect, and
Definitely, definitely. No,
that's what's important.
yeah,
And
that's
the
the yeah.
Novavax is it's great at what was it 94% protective, but the question now becomes you're the third. We have enough vaccine where one of the worries in the Philadelphia area right now is that there's going to be about 4,000 doses of the J&J vaccine that are simply going to be thrown in the trash because they made it up and nobody's showing up to get the vaccine. So it's going to be the same thing.
Yeah,
Is
but if
is
the Novavax is the best, it protects from the variants, from the mutation of the Delta variant, and also if there is less side effects, I think that the next vaccine will definitely be Novavax.
Well, but they're saying that the Moderna vaccine protects against the Delta variant also. And so,
Mm.
is there a difference between
But it
94%?
has side effects, Moderna. I have at least in Europe, they do not love Moderna a lot because of its side effects. People get sick.
Yeah, I mean, I had the Moderna, and yeah, I didn't run a low grade fever, but my arm was sore for 5 6 days
That's for
afterwards.
all of them, for all of
For
them.
all of them, exactly.
Yeah.
And you know what? In fact, that's an interesting point about the side effects is that in the clinical trials, people are used to when they get a vaccine that they're going to see a side effect, that their arm's going to be sore, they're going to get a low grade fever. And what the anecdotal reports are coming across the wires in this country, at least, was they were seeing a lot of dropouts in
Mhm.
the clinical trials for the vaccines because people were not getting soreness in their arms. They weren't running low grade fevers and they figured, oh, I didn't get the vaccine. I got the placebo.
Mhm.
So they dropped out of the vaccine trials and they went and got themselves enrolled in another vaccine trial, which engendered another area of statistics of dealing with missing data.
Mhm.
You started with a million patients and now you don't have a million patients any longer. How do you prove the efficacy? when
Mhm.
you've got all these patients that are hopping between one trial and the next, not realizing how
Mhm.
long it's going to take to raise the neutralizing antibodies to prove that the vaccine really works. So, the value of something like the Novavax may be for far emerging countries where you haven't had enough of the Moderna or the J&J or the Pfizer vaccine to be able to address all their
Yeah, I
needs.
read in the same article written by this European professor, I said that Europe is really expecting, a rapid distribution of Novavax, at least for September. So he was really excited by knowing that Novavax probably can reach the market by that time. I also read that they have run their clinical trials in Peru and Colombia in South America, where the situation was really very intense from COVID infection. And there were unknown variants, that today we do not have it yet, at least. Thanks God, not in Europe. I hope
Yeah.
that it will never reach to anybody. So it was a very good example. So, it is hopefully probably even from the upcoming variants, it has a very good efficiency rate that can protect a lot of. I think
But, but
that, yeah.
there was this article on the wire that somebody sent to me local recently. I'll see if I can find it again. Comment by Luc Montagnard, the Nobel
Mhm.
laureate French scientist from the Pasteur Institute who worked on
Mhm.
HIV along with Bob Gallo, and he claims that the variants are being produced by the vaccines,
Oh no,
and that is
really?
being picked up by the anti-vax groups
Oh
and the
yes,
vaccine hesitancy people saying, here's why you shouldn't get vaccinated because the vaccines are producing. Now, my retort to that is, if indeed the vaccines are producing variants, then how did the Delta variant occur in India where they have an incredibly low vaccine rate? And why didn't the variant occur in Italy, in Israel, where they have a very high vaccine rate?
definitely, yeah.
So his logic doesn't hold up at this stage, but it's being picked up and being used as, we would call it, cannon fodder for people to say, here's why I shouldn't get the vaccine,
Okay.
is because it's going to engender more variants popping up. So
Mm.
it's going to be more than just the UK variant, the South African, Brazilian, and the Delta variant from India at this stage. So, this is the problem with this disinformation, is
Mhm.
that you hear what you want to hear and you use the information as you wish it to be used.
Definitely, yeah, yeah, that's true, that's true, and that's a big problem. And what about, the next generation of the vaccines, COVID vaccine, probably that I think one of the Indian companies, Bharat, is working on a vaccine that it's nasal
Intranasal,
spray. Yes,
yes.
intranasal. What do you think about it? Do you think it will be as efficient as the other vaccines? And is there any
It's going to be interesting.
chance?
Efficacy is going to be interesting. It has a niche, and part of the niche is for people who are needle hesitant.
Mhm.
It also has the advantage of for mass introduction to
For children,
populations.
also for children
Well,
and young
that's been
adults.
tried in this country. There was a company, MedImmune,
Mhm.
in this country, which is really part of AstraZeneca,
Mhm.
had a vaccine for influenza called FluMist,
Mhm.
and it was specifically targeted at children,
Okay.
figuring out that children have trouble with needles and parents have trouble watching their kids get injected, and it was introduced into the market and it was totally ineffective. It was so bad that in fact pediatricians recommended don't use it,
Okay.
as did the CDC. It's gotten better, but it is still a niche product. But for use in far emerging countries where you could go, door to door with something like this and
Okay.
vaccinate large numbers of people, what will have to be seen is if that vaccine, because it's being developed in India, but it's also actually, I think, it was developed somewhere else. I find it
Mhm.
because they have a website. The
Mhm.
company that's developing it was not the originator.
Yeah. Yeah. Yeah.
It
Yeah.
was done
Yes.
partly, I
You're
think,
right.
by the
Mhm.
National Institutes of Health and a
Mhm.
university. It may
Mhm.
have been
Yeah,
University of
that's
London
true. That's true.
developed this,
Mhm.
and so they're co-marketing it because it's the same problem they've had with vaccinating for polio in India.
Mhm.
Is
Mhm.
going door to door with little foam chests filled with refrigerated vaccine, and nasal vaccine may be much
Mhm.
better.
Mhm.
But the nasal route's been looked at a lot for a number of drugs as a route of administration. The problem will be is making sure you get enough into the nasal mucosa, get enough absorption,
Mhm.
and that you have enough of a load that whatever is in there. I think they're trying to generate IgA and G antibodies, is that it doesn't run down the back of your throat, doesn't run down, dribble down the front of your face, and that
Mhm.
it stays there long enough. So again, it's going to be an interesting delivery technology that's being looked at for other types of drugs. People looked at it for, I think GSK at one point was looking at it for their migraine drug. And they figured, if you have a migraine, one of the things you can't tolerate is swallowing something.
Mhm.
You're nauseous, you may vomit, and you may vomit the pill. And so they tried it for their migraine drug, and the problem became dosing.
Mhm.
of and how are you assured how much you actually got into the system by spraying it into somebody's nose? How do you compensate for what dribbles down the face and what goes down the back of your throat?
Mm-hmm.
So for vaccines, it may be less necessary, but you're going to have to figure out what kind of contact problems are you going to have with the vehicle
Mm-hmm.
that the vaccine is in. And does it produce irritation of the nose? Is there enough nasal mucosa to get enough absorption of the material?
Mm-hmm.
How do you test for safety
Mm-hmm.
for something like that? You can, injecting into an animal, you know what's going to go on. You
Mm-hmm.
know about tenderness in the site.
Mm-hmm.
What happens when you do that into somebody's nose? Do
Mm-hmm.
you get adequate? It may be a perfect solution for developing nations, be
Mm-hmm.
able to do mass vaccination clinics, very similar to what was done when the Salk vaccines came out and then the Sabin vaccine, where they would line kids up, put a drop of the vaccine onto a sugar cube, and stick it in the kid's mouth and tell them go home.
Mm-hmm.
Um, this may be a
And
very valuable technology for far emerging countries if
mm-hmm.
it holds up, and if there's durability for the vaccine. This is the immune response going to last as opposed to being injected, and that was the objection from the World Health Organization and Johns Hopkins to the Novio
Mm-hmm.
technology
Yeah,
of
yeah.
using
Mm. Yeah, yeah.
electroporation.
Yes,
It
yeah,
just
yeah.
didn't lend itself
It's
to
very
mass use,
complex.
mass
Mm.
vaccination
It's just
clinics.
impossible yet. Yeah, that was my last question. Yeah, is there any technology platform that is not yet used for a COVID vaccine? Something promising that could exist or is not yet used for the COVID vaccine. Is there anything?
What I think people are looking at, not necessarily for COVID, although it would be important, is continuous
Sure,
manufacturing of
sure.
vaccines.
Mm-hmm.
There are only three drugs, I think, approved in the United States, small molecules. Vertex has one that are being produced by continuous manufacturing.
Mm-hmm.
But continuous manufacturing of biologics is the hot area that the Food and Drug Administration and the EMA are very interested in because that would allow you to rapidly produce a vaccine,
Mm-hmm.
and that would be key to getting rapid deployment instead of having to make a huge factory at a
Mm-hmm.
cost of $3,000 a square foot to be able to crank something out, so to say, of
Mm-hmm.
having disposable bioreactors, single use, as some small companies are doing right now. Sartorius makes a lot of this single use material, Accelerex, single use technology, up to 2,000-liter bioreactors, which you could need. And you could establish a facility someplace very, very quickly, but they're not quite at continuous manufacturing. They're simply at the stage where instead of having to have a 20,000-liter bioreactor that Lonza has or 30,000 liter in Singapore, you'd have a 2,000-liter bioreactor. You simply would run it 10 times to get the same volume with the associated risks of managing the technology. But that may be the key. So a lot of people are looking at the vaccines of the future of being in the position of doing continuous manufacturing. Now, people have looked at that. They used to call them chemostats 40 years ago, and you would make up bacterial cultures and do it that way. The technology now is to be able to think about doing that for a vaccine. That may be the key to getting new vaccines out there and
Mm-hmm.
multiple copies of vaccines, different types of vaccines to address the issue of variants. This SARS-CoV-2 vaccine's been interesting. With all these variants appearing. What's the environmental pressure that's
Mm-hmm.
producing these variants, and so widely distributed? Brazil versus South Africa versus India versus the UK variant. What are the drivers evolutionary? Are we learning anything
Mm-hmm.
from these? Because I've seen a map. The Economist had one about 2 months ago of all the variants, and there's lots of variants out there. Not all of them are variant enough
Mm-hmm.
to warrant being concerned about them. Now that the Delta variant's out there, it's more infective,
Mm-hmm.
potentially more severe disease. So
Mm-hmm.
that's why the concern of what's happening in India. But they've only vaccinated, was it 5-6% of their population? It's really a problem. They were at the forefront of managing, and all of a sudden they got sloppy. It's the best way to say it. And now you've got all these variants appearing. So continuous manufacturing may be the way to rapidly address the occurrence for the next pandemic. Uh, but what's going to be important too, and we're really falling down on this, is what are the vectors? I guess everybody's all upset over World Health. They're upset over the Wuhan, Institute. Was it really a bioengineered virus that, I won't say it leaked? The Chinese laboratory has had issues with biosafety.
Mm-hmm.
It's a BSL-4. That's what we would call a Andromeda strain. Um, so somebody touched something and they carried out. Nobody touched anything. If you ever seen pictures of people in a BSL-4 lab, they're in these huge inflatable suits. Nothing is
Yeah,
touched and carried out
it's
of that
nothing.
laboratory.
It's
It's
just
negative pressure.
impossible. Mm-hmm.
The rooms that you down, when you leave, you are washed down and disinfected before you even get out of that suit. So unless it was intentional, that's not a solution. But we don't know what the vector is, and everybody's upset about that. Well, guess what? We don't know what the vector is for Ebola, and Ebola's been around for 50 years. We
Mm.
still really don't know for sure what the vector was for SARS-CoV-1, and that's still present. We sort of know what the vector is for MERS, camels, and yet it's a real significant problem in South Korea. And last time I checked, there aren't any camels in South Korea, but it's still a problem there, and it's got a 40% mortality rate. It's a much more infectious coronavirus.
Mm.
Now there are people talking about maybe we need a vaccine for SARS-CoV-2 in animals because there are reports of it, being present in cats. There have
Mhm.
been reports of people finding it in zoo animals.
Mhm. And can they transfer it to human
That's a
cats?
really interesting question. Is
Mhm.
people are watching to see, do you get that jump from, say, zoo animals to their keepers when they suddenly test for a COVID-2
Mhm.
positive tiger
Mhm.
in a zoo? I don't think people have quite looked at primates in zoos yet because we don't have that many primates where a primate in a zoo is not something you want to come in contact with. You don't exactly sit around and have a cigarette with a chimpanzee. It's not the sort of thing that people do. They'll tear your arm out of the socket. But um, it's a valid question. I think people are beginning to really question is that should we now be vaccinating our pets? Because for something like a cat that goes outside and contacts other feral cats,
Definitely, yeah. Mm.
and that's an issue in the States where we don't vaccinate cats as frequently as we should for rabies. And
Okay.
cats don't get rabies, but cats can carry rabies.
Mhm.
So that's a problem when you get scratched by a cat. In fact, just last week in this area, maybe 30 kilometers south of where I live, a young girl was attacked by a fox.
Oh,
And
okay.
it turns out that fox had attacked two other people. And
Mm.
so the fox was trapped and killed, and now they're testing it for rabies.
Mm.
But, I was reaching a point where should we start testing some of these feral animals for COVID? And it's a worry, but can you worry about everything, or should we be really planning for that next generation of whatever else is out there, and learn? As we talked about, we're not a learning society, and this will go by its wayside. We'll get it control, although we just hit this milestone yesterday. 600,000 people have died. Over 3 million people have died worldwide, and not an awful lot of people are still upset over it. We still have these people here who claim it's no worse than influenza, and they are pushing back over vaccinating teenagers. And in fact, there was just an article in one of the local newspapers written by two pediatricians objecting to vaccinating children. It's a benign disease in kids; they don't get all that sick. Flu-like symptoms. And why are we spending all this money doing clinical trials now on 6 and 7-year-olds where it is not that much of a health burden on these kids, but not thinking about the fact that those asymptomatic kids can then bring it back to their grandparents who may or may not get vaccinated. And even if they have been vaccinated, that 94% is probably for the general population as a whole. It's not for people my age. Unless somebody's starting to do these long-term studies on elderly or the truly elderly in Japan, where you have people 90 years of age, when do you see breakthrough? How long is the vaccine going to last? When do you need to
Mm.
boost people over 70? In all of our populations, look at the problems Italy had because they have such an aging population. That was the big thing that crashed the Italian healthcare system, where all these people over 65 and 70 that really produced a burden and were really dying, at
Mhm.
an extensive rate before the vaccines were available, and they were just ill-equipped to deal with that patient population in their hospital settings. They even ran out of the valves on the respirators, the ventilating machines, and there was a company that stepped forward and broke the patent of the company who made the valves and started 3D printing them and offering them for basically $1 to local hospitals in northern Italy because
Mhm.
that was a rate limiting step. Was you needed to have those things. They needed to be autoclaved, and they didn't have enough valves for the ventilators. So this, I think, is changing a lot of things. That was that people looked at it and said that's open innovation. Here's a small company. They saw a need. The company that made the valve, which was $300 a piece,
Mhm.
couldn't supply them fast enough. And this small, university company said, look, here, we can 3D print these things. They're different from a color standpoint, and they
Mhm.
started cranking them out. So here was open innovation.
Mhm.
But there's a book I have on my shelf. I'll send you the title. It's called The Little Black Book of Innovation. And it
Okay.
goes along with an article that appeared in the Wall Street Journal 3 years ago that said, enough innovation, just get something done already.
Yeah, definitely. Now, thank you very much for this very insightful discussion, Dr. Gross. I
I don't
just
know how
got
insightful
the
it is. I just enjoy the chats with you because, perceptions of what
no,
innovation is,
it is
what's
very
happening next. Across the pond, we have different attitudes towards some of these things. And, who funds the research? As and how the research is done, as my dean calls it, one always has to be cognizant in the United States of dealing with what we call OPM, other people's money.
Mhm.
And how do you use it wisely, and how do you really make it meaningful? And then this dichotomy in the United States of basic research versus applied research. And that's a problem, and that's the issue in universities is trying to do good applied research, trying to develop that next platform is tough to do at universities because your job is there to discover the truth. And I'm sure you face the same thing at your university and many other universities. It's a tough road to haul if you think you've really got a great idea for a new platform that's going to either be a new medical technology device or better a test, a more deployable test, a cheaper test. Um, but thinking about the entire ecosystem is very important. In this country, at the height of the epidemic last year, our public health service was very proud of the fact they were rolling out millions and millions of home test kits that could be sent in an envelope. It was a cardboard card. You stuck a sterile swab that came with the kit into your mouth. You swabbed your nose, I guess, and put it on this card. And if it turned purple, you had COVID-19. What they neglected was, who did you tell? How did you get the data to anybody? So
Mhm.
you were positive. Did you call your county public health department? Did you
Mhm.
call the CDC? They forgot about the fact that if you got the data, what did you do with it? So I think this has created that it's forced us to think about this entire ecosystem of managing the disease. It's
Managing, yeah,
managing it. It's brought
and
as
managing
word
innovation.
managing innovation, making it, you hate to say useful,
Yeah,
but the
useful.
deployable innovation. If it's going to cost, a bloody fortune, it may not be deployable. And then how do
Definitely.
you deal with the
Mhm.
data? I'm a fan of big data, but unless it is actionable
Mhm.
information, who cares? If it doesn't help somebody, make a decision.